Merci. Je vais commencer avec un peu de français. D'abord, je veux remercier toute l'équipe de MATHEC, et spécialement Dominique, pour me donner l'opportunité d'être ici. C'est vraiment un privilège pour moi d'être ici pour beaucoup de différentes raisons. Mais ce que j'essaierai de faire, c'est de discuter avec vous deux choses qui ressemblent très simples, mais vraiment ne sont pas assez simples. Une, c'est la définition des MSC, et l'autre, c'est comment est-ce que cette cellule fonctionne in vitro et un peu in vivo. Alors, j'espère que vous me pardonnez, mais je vais changer ma langue à anglais. Mais s'il y a quelque chose que je n'explique bien, s'il vous plaît, dis-moi et je vais essayer de l'expliquer en français. At the lab, when we think about mesenchymal stromal cells, what we think immediately is about the definition that was proposed in 2006 by the International Society for Stem Cell Therapy. And that implies that these cells have a fibroblast-like morphology, that they are able to differentiate into all skeletal tissues, and that they do have certain surface markers which are there with the green isotype, CD90, CD73, and CD105, and they do not express other markers that we expect to see in hematopoietic lineages. However, and I really want to emphasize this, the reason why these criteria were developed was to streamline research in this area in order to have a common language of what we were calling MSCs. These criteria were not developed for clinical purposes, and I think that's really important. This is not enough to say that an MSC is functional and will be functional in vivo. Now, the second concept that I would like to highlight here is that not every MSC fulfills these criteria. In fact, for example, if you isolate MSCs from adipose tissue at an early passage, those cells do express CD34, or a fraction of them do express CD34, and then after you pass them, that marker is lost. The third thing that I want to emphasize here is that this concept of the capacity of these cells in vitro to differentiate into different lineages was what led the idea that, in fact, the function of these cells was secondary to the same properties in vivo. So these cells would have been able to differentiate in vivo and, in that sense, replace function. However, I'm going to show you data that really tells us that this concept is not correct. So where do we find these cells in vivo? If we think about the composition of an organ has essentially three types of cells. One are the parenchymal cells. So those are the cells that really do have the main function that relates to the organ that we are thinking about. The second one are the external linings. For example, in the case of the heart, we think about the pericardium, or in the case of the brain, the meninges. And then there's a third part of cells, which is what it's called the stromal vascular fraction. And again, when we think about stromal vascular fraction, there are different cells that are there. One of them are the mural cells, and among those cells, there are two subtypes. One are pericytes, and the other one are vascular smooth muscle cells. Currently, we think that MSCs are pericytes, and those pericytes essentially surround small caliber vessels, and what they do is they stabilize those vascular networks. What happens in vivo? This is a recent study that I unfortunately do not expect you to see all the details of the images, but I'm going to tell you about this. This is the first study that was done and was able to follow the fate of pericytes or MSCs in vivo. And that's why, despite of this being an animal study, I'm showing this to you, because this is something that we would not be able to have proof in humans. So the trick here is that the authors have found that there is a specific transcription factor, which is called TBOX18, which is only and specifically expressed by pericytes. So then they were able to generate animals transgenic for this transcription factor, and they looked at where, in fact, these cells are located. The bottom line of this image that I wanted to show here is that these cells are located essentially in every single tissue of the animal, but the distribution of those cells and the way they are organized differs from one organ to the other one. In some of them, they form a line. In some other organs, they are scattered within the interstitial cells, and in other organs, they are in the medial layer of that organ. The next step that these authors have done was to isolate these T-box18 positive cells. And in vitro, they induce them to differentiate. And in fact, these cells do differentiate into the 3 lineages that we expect an MSC or a parasite would differentiate. So that is fine, and that's good. But the next step that they have done, which is really where the core of this work is relies, is that they have generated flox animals transgenic with the T-box18 plus 3 different models of injury. So lung injury, heart injury, and brain injury. And also they have looked at the effect of aging, and also they have looked at animal models in which they fed with a high concentration of fat. And they followed those animals throughout time. In fact, the aging model was a model that they followed for 2 years, which is huge. In these models, they were able to prove that, in fact, parasites were not multipotent progenitors. They are not able to differentiate into adipocyte progenitors or fibroblast progenitors or neuron progenitors. And in fact, the in vitro plasticity of the MSC was shown to be an artifact of what we do in vitro, but not something that occurs in vivo. So really, this puts away the idea that MSCs are mesenchymal stem cells, because in fact, to be a stem cell, that cell needs to be able to differentiate in vivo into multiple lineages. Despite of that, what we really know is that these cells are used for the last 15 years. I'm just showing you the first paper that is a case report that used these MSCs for a patient that had GVHD, acute refractory GVHD. And that's not, of course, the only disease in which these cells are used. You have heard about the trials that are currently running here by Dr. Farge in lupus and in scleroderma. And these cells work. So no matter how we call them, these cells do have an impact in the way the immune system works. However, if I have to go into detail and tell you exactly what are the mechanisms of action of these cells, I will not be able to do that. And what I'm going to show you is things that we have learned and that may change within the next few years. So the first thing that we know is that there are certain active mechanisms that account for the mechanisms of action of MSCs, and there are certain passive mechanisms. The active ones is the capacity of these cells that fuse to other cells, and this has been shown in the heart, the capacity of these cells to produce paracrine factors and modulate other neighbor cells, the capacity of these cells to establish tubules and transfer contents from the cells through these tubules, and the capacity to produce extracellular vesicles. Those are the active mechanisms. The passive mechanism is the fact that these cells are phagocytosed by other cells, and then those cells are the ones that are reprogrammed and have an effect. As a consequence of all these different mechanisms of action, we know that these cells have the capacity to modulate the immune system, but also that these cells do have pro-angiogenic effects, and they do modulate fibroblast function with a net antifibrotic effect. So what happens when we use MSCs in vivo? This again is something that would not be possible so far to do in humans. This is a model in which umbilical cord MSCs were stained with two different dyes. One allows to look at live cells, and the other one allows to look at dead cells. And what they have done was to look at what happened 5 minutes, 24 hours, and 72 hours after the infusion. The concept here is that within 24 hours, all MSCs that were injected in this animal were cleared. At 5 minutes, we see that most of the cells are in the lungs, where we think the cells are trapped. And then at 24 hours, all the cells are not longer live, they are dead, and they localize mostly in the liver, where we think are cleared by Kupffer cells. So how is it possible that a cell that disappears 24 hours after we administer it does have long-term effects? So the model that this paper proposed, and there are experiments that they have done to prove this, which I'm not going to go through, is that after the injection of these MSCs, the cells go into the capillaries of the heart. They are phagocytosed by local innate immune cells that then are redistributed through the blood, and there they're able to change the phenotype of monocytes from a non-inflammatory phenotype, from a pro-inflammatory phenotype to a non-inflammatory phenotype. And ultimately, then these cells localize into the liver, where they are cleared. So what these data suggest is that, in fact, the main effectors of the MSC function are cells from the innate immune system, phagocytes, specifically neutrophils, and monocyte macrophages. And then those cells are the ones that are going to educate cells of the adaptive immune system, which leads to the ultimate tolerant effect that we see from these cells. One of the concepts that I think it's really important for us to understand is the fact that for an MSC to have an effect, that MSC needs to be licensed or primed. And that occurs because these cells have a unique capacity not only to modulate their environment, but also to sense their environment. How do we know that an MSC is primed? Essentially because these cells baseline do not express HLA-DR, and after they see interferon or TNF, for example, they do increase the surface expression of HLA-DR. So again, the first concept here is that MSCs are not constitutively immunosuppressant, but they become according to where they are exposed. What I'm going to show you next is just a model. This, we do not know whether it happens in vivo, but what it is proposed is that MSCs can polarize depending on what is the condition that they sense. So if they sense high levels of interferon gamma and TNF, or if they get in contact with double-stranded RNA, which triggers TLR3, that would induce the generation of what are called MSCs type 2, which are capable of have an anti-inflammatory effect. In contrast, if the same MSC is in an environment where there are low levels of interferon gamma or TNF, or there is, for example, activation or stimulation of TLR4, that MSC would become a pro-inflammatory phenotype. So how do we understand this? It's just thinking that if there is a starting of an infection, that MSC needs to alert the rest of the immune system that they need to become activated in order to get rid of that infection, whereas in fact, in the presence of a highly inflammatory environment, the MSCs needs to do exactly the opposite and needs to try to calm down that fire. So again, the concept here is that the MSC response varies depending on what is the nature of the immune stimulus and also what is the response that the immune system has to that particular stimuli. What we do know is that the effect of MSCs does not depend on the cell-cell contact of MSCs with the immune cell. And in fact, one way in which we look at this is with co-cultures of T cells that are in the presence of MSCs, and they can be in direct cell-cell contact or not in cell contact if we put them in a trans well. And what we know, and this is of course something that it's not new, is that the effect of the MSCs can be, in more than 80% of that effect can be reproduced if the cells are not in cell-cell contact. So what that means is that there's something that these cells produce that can regulate the function of the second cell that is soluble, and it's in the secretome of the MSCs. This is essentially one of the main functions of the MSCs, which is the paracrine activity. And what I'm showing you here is that when we do have activated CD4 T cells and we put them with MSCs or with the secretome of MSCs, we do see after they are primed that there is a decrease in the proliferation of CD4s. And that relates to multiple factors that have been described up to now. One of them is a decrease in IDO, but also a change in the relative proportion of pro-inflammatories and anti-inflammatory factors. And again, the amount of IDO and of kynurenine, also the amount of IDO and the amount of different pro-inflammatory factors change depending on what is the activated state of the MSC. The last mechanism that we have investigated is the capacity of these cells to establish what are called nanotubules and to connect with other cells with a unique possibility of transferring organelles from the MSC to other cells. So these are the TNTs. They are essentially prolongations of the cytoplasm of the MSCs. And you cannot see here, but believe me, there are specific red dots that are essentially mitochondria from MSCs. Why this is important? Because we know that this can be a rescue, a way in which the MSCs can rescue another cells through the providing those other cells mitochondria that are then able to give an OXPHOS signal and induce the production of ATP by the receptor cell. And this is a mechanism that specifically has been shown to be relevant in acute respiratory distress syndrome. So I do believe that there are many things that we don't know about these cells, but there are certain things that we know, and I think they are important to remember in order when we think about translating these cells into clinical practice. The first one is that we do not expect that MSCs will be in the recipient forever. These MSCs are going to be cleared for the system of the recipient within 24 hours. They have a short therapeutic activity. They have a short window of therapeutic activity, but the effect persists afterwards. We do not expect that these cells will home into the site of injury, but despite of that, again, these cells do have therapeutic efficacy. They need to be activated by the inflammatory environment. They do not respond in a monolytic way. That means that it's not that an MSC will always behave in the same way, but it will depend on where we administer that cell. The primary effect of these cells, how we understand them currently, is that their effect in the innate immune system and afterwards in the acquired immune system. And the last point is that there are several mechanisms of action that these cells have. They are not mutually exclusive, but one of the key consequences of that is the capacity of these cells to regulate the immune system. This is the group of people that work in the lab. We are really looking forward to have Alice with us. These are the collaborators that really provides the samples that we use at the lab, which are all samples from humans, and these are the companies that fund our work. The last thing that I wanted to say is that I really want to say a special thank you to Dominique, and when I was thinking last night about her, I thought that really the three things that I was able to appreciate since I know her is the limitless energy that she has, the strength, the dedication, and the generosity, but more important, her vision. And as Marie said, this really made me very proud to have had the opportunity to meet a person and consider her a mentor with such a vision. Thank you so much.